Hello students, welcome to EPG Pathshala. I am Dr. Janvi from the Department of Biochemistry, AIIMS, New Delhi. Today we are going to talk about the module Nucleocytoplasmic Interaction and Cell Function. This module is placed in paper Cellular and Molecular Biophysics. The genetic material or DNA of all eukaryotic cells, whether they are animal cells, plant cells, protists or fungi, is stored in the nucleus. This nucleus is enclosed in a double-layered membrane called the nuclear membrane or nuclear envelope. This nuclear envelope separates the nucleoplasm from the cytoplasm and controls the nucleocytoplasmic transport, which is critical for many physiological processes, including gene expression and signal transduction. It is also implicated in pathological cell processes such as oncogenesis. By establishing a nucleus, eukaryotes compartmentalize and separate transcription from translation. This enables the process of gene regulation to take place. This distinguishes eukaryotes from prokaryotes. It is estimated that a human cell can transport up to 80 megadaltons of material per second across the nuclear membrane. The nucleocytoplasmic transport of macromolecular cargo is regulated by a complex interplay of nuclear transport machinery, which includes elegant structures like nuclear pore complex, nuclear envelope proteins, and various soluble transport receptors. In this module, we will first discuss the roles of nuclear membrane in establishing this interaction between cytoplasm and nucleoplasm, followed by an illustration of the traffic controller of the nuclear membrane, a key structure known as nuclear pore complex or NPC. Based on the structural assembly of NPC, several models have been proposed to explain the selective permeability barrier of nuclear pore complex. We will discuss 4 of these proposed nuclear transport models. We will also deliberate about the factors affecting nuclear transport and the nuclear transport cycle. Finally, we would understand how nucleocytoplasmic interaction is important for cell function by studying some diseases caused due to defective transport across nuclear membrane. The nuclear envelope is a physical barrier between nucleoplasm and cytoplasm. It is composed of a phospholipid bilayer membrane which consists of 2 layers, the inner nuclear membrane and the outer nuclear membrane. These inner and outer nuclear membranes are separated by a perinuclear space. As can be appreciated in this picture, the outer nuclear membrane is in continuity with the rough endoplasmic reticulum, which is studded with ribosomes engaged in protein synthesis. The nuclear membrane is perforated by nuclear pore complexes. These nuclear pore complexes fuse the outer nuclear membrane and inner nuclear membrane together and creates channels for nucleocytoplasmic transport. As seen in this picture, nucleus transports materials to the cytoplasm, including mRNA, tRNA, and ribosome subunits. It also imports materials like enzymes, nucleotides, and regulatory proteins from the cytoplasm across the nuclear pore complex. In this picture, you can see a nuclear pore complex assembly in red, placed at the fusion of inner and outer nuclear membrane. You can also see that the inner membrane is attached to a meshwork of intermediate type 5 filaments called nuclear lamina. In mammals, 3 types of nuclear lamins line the inner surface of the nuclear inner membrane: lamin A, lamin B, and lamin C, as seen in this picture. The lamins bind to specific proteins present in the inner nuclear envelope such as emerin and lamin B receptors. The lamins provide skeletal support to the nuclear envelope. These also provide the site of attachment for chromatin fibers. Lamin also help in disintegration of nuclear envelope at the time of cell division and its reorganization after the cell division is over. Mutation of lamins or lamin-associated inner nuclear membrane proteins can result in tissue-specific diseases known as nucleoenvelopathies. Mutation in lamin A leads to Emery-Dreifuss muscular dystrophy, whereas mutation in lamin C leads to limb-girdle muscular dystrophy. Nucleopore complexes, also called NPC, are multi-protein suprastructures with sizes of 50 to 112 megadaltons that allow exchange of matter, energy and information between two major compartments of the cell, the cytoplasm and the nucleus. The individual transport rates of NPCs are very high, about 1000 cargo transport complexes per second. All the histones and ribosomal proteins needed per cell cycle are transported through the NPC. NPCs within nuclear envelope display variability in density and distribution between cell types. In majority of somatic cells, the nuclear pores are randomly distributed on the surface of the nuclear membrane. But sometimes the NPCs can be arranged in hexagonal symmetry, as in the Malpighian tubule cells of grasshoppers, or in clusters, as in the oocyte of Xenopus laevis. In this picture, you can see a nucleopore complex assembly in red, placed at the fusion of inner and outer nuclear membrane. You can also see that the inner nuclear membrane is attached to the meshwork of intermediate type 5 filaments, called nuclear lamina. In mammals, 3 types of nuclear lamina line the inner surface of nuclear inner membrane: lamin A, lamin B and lamin C, as seen in this picture. The lamins bind to specific proteins present in the inner nuclear envelope such as emerin and lamin B receptors. The lamins provide skeletal support to nuclear envelope. These also provide the site of attachment for chromatin fibers. Lamins also help in disintegration of nuclear envelope at the time of cell division and its reorganization after the cell division is over. Mutation of lamins or lamin-associated inner nuclear membrane proteins can result in tissue-specific diseases known as nuclear envelopathies. Mutation in lamin A leads to Emery-Dreifuss muscular dystrophy, and mutation in lamin C leads to limb-girdle muscular dystrophy. Nucleopore complexes or NPC are multi-protein suprastructures with the size of 50 to 112 megadaltons. These structures allow exchange of matter, energy and information between two major compartments of the cell, the cytoplasm and the nucleus. The individual transport rates of NPCs are very high. There are about 1000 cargo transport complexes per second. All the histones and ribosomal proteins needed per cell cycle are transported through the NPC. NPCs within nuclear envelope display variability in density and distribution between various cell types. In majority of somatic cells, the nuclear pores are randomly distributed on the surface of nuclear membrane. But sometimes the NPCs can be arranged in hexagonal symmetry, as in Malpighian tubule cells of grasshoppers, or in clusters, as in the oocytes of Xenopus laevis. In plants, protozoan and amphibian oocytes, NPCs account for 20% of total nuclear membrane area. However, in mammals, NPCs account for 5 to 15% of the total surface area of nuclear membrane. This NPC density is determined by transcriptional activity of the cells. More active cells have high number of NPCs. For example, non-dividing RBCs and lymphocytes have nuclear pore density of 3 per micrometer square, whereas highly active cells like hepatocytes or brain cells have nuclear pore density of 15 to 20 micrometers per square. About 3000 to 4000 NPCs are present on a mammalian cell nuclear envelope. While small-sized molecules less than 30 kilodaltons move freely through the nuclear pore, bigger molecules require nuclear localization signal, also known as NLS, and a soluble transport receptor to mediate transport across NPC. Nuclear pore complexes are elaborate and complicated protein assemblies. NPCs form a diffusion barrier for certain molecules and simultaneously remain highly permeable to other molecules. This remarkable selectivity is accomplished by the structural organization of NPC. NPC morphology is conserved from yeast to humans. Using cryo-electron microscopy and cryo-electron tomography, NPC structure with resolution up to 6 nanometer has been delineated. In vertebrates, NPC consists of 600 individual proteins forming a structure with a characteristic 8-fold rotational symmetry. NPCs from various eukaryotic origin share a set of 30 nucleoproteins. These nucleoproteins, also known as NUPs, are often grouped into 3 categories. The first category is the transmembrane NUPs, which anchors the NPC in the nuclear envelope. The second category is the FG NUP, which forms 1/3 of all NUPs and contains phenylalanine glycine repeats, that is FG repeats. The third category, structural NUPs, consists of half of all NUPs and forms a scaffold with transmembrane NUPs and FG NUPs. In this picture, you can see the donut-shaped nuclear pore complex. This donut-shaped nuclear pore complex is made up of 3 lattice-like ring structures termed cytoplasmic ring, CR, shown in purple towards the cytoplasmic side, the nuclear ring, NR, shown in purple towards the nucleus side, and the inner ring complex, IRC, in between the two. The 3 rings are sandwiched between cytoplasmic extensions, shown as red filaments extending into the cytoplasm, and nuclear extensions. The nuclear extensions, which extend 60 nanometer into the nucleus, are linked by a distal ring to form a basket-like structure called the nuclear basket, which can be appreciated in green color here. Between cytoplasmic and nuclear rings is the vertical structure called central framework. This central framework harbors the central transport channel that accommodates all active transport across NPC. The NUPs or nucleoproteins are organized in small set of subcomplexes which are defined by the stability of protein interactions. The largest subcomplex of NPC is the Y complex, named after its Y-shaped structure as seen on the right. In its simplest form, the Y complex has 7 proteins in yeast and 9 in vertebrates. The Y complex is the main component of cytoplasmic and nuclear rings. These rings are involved in various important processes like mRNA export and DNA repair. The Y complex is an essential scaffold unit within the NPC, depletion of which abolishes the nuclear pore complex formation. In the picture on the left, cryo-electron tomography reconstruction of the nuclear pore complex has been done for yeast and metazoan cells. Nucleoporins from yeast and metazoa are listed and color matched according to their approximate positions within the NPC. We can see that the nuclear basket, which is marked in green, is made up of NUP1, NUP2, NUP60 and MLP1 and 2 in yeast. The regions of FG NUPs are known to be intrinsically disordered, and a lack of secondary structure happens in FG NUPs. These FG NUPs regions are flexible and allow the protein to adopt different conformations in a short span of time. FG repeat domains can interact with many different nuclear transport receptors, also known as NTRs, to adopt a defined structure based on the protein-protein interaction. Based on structural insight, several models for selective permeability of NPC have been suggested. The first one, shown in this picture as A, is called the selective phase or hydrogel model. In this model, the diffusion barrier of nucleopore complex is a three-dimensional sieve that allows movement of small molecules but restricts that of larger ones. The molecular sieve is formed as a result of intra- and intermolecular interactions of cohesive hydrophobic clusters within FG NUPs. A gel-like network is formed with the mesh size defined by concentration of FG NUPs and distance between individual FG repeats. This model explains the facilitated diffusion of NTRs and restricted influx of inert molecules. It also explains how NTRs and transport complexes can overcome the sieve-like barrier by transiently interacting with hydrophobic FG clusters. A physical sieve with defined mesh size also explains the size limit for passive transport across NPC. However, it has been observed in single molecule study experiments that cargo-free or cargo-bound nuclear proteins travels at the periphery of the nuclear pore. These observations may be explained by another model of nuclear transport. The second nuclear transport model, shown as B, is the virtual gate or polymer brush model. This model is based on thermodynamic considerations. The movement of molecules from large cytoplasmic or nuclear volume to a small volume of the NPC causes a loss of entropy. FG filaments form repulsive bristles or polymer brushes, creating an entropic gate. At the same time, interaction of molecules with FG repeats will lead to release of binding energy, resulting in change of enthalpy of the system. If the binding energy is high enough, it may compensate for the loss of entropy and allow the molecule or NTR transport complex to move across NPC. The third nuclear transport model marked in C is the forest model. FG domains can form two distinct conformations, a globular collapsed coil conformation, which are known as shrubs, and a collapsed coil with extended coil conformation known as trees. Together, a forest-like landscape is formed by alternating trees and shrubs to fill the volume of the nucleopore complex. Collapsed coil domain of several trees converge in the center to form a zone 1, whereas the peripheral zone 2 harbors extended coils. Zone 1 acts as mesh gate and zone 2 as entropy gate. This model explains travel through the center or transporter region of NPC as well as passage through the periphery of NPC. The fourth model is shown as D and is called Reduction of Dimensionality or ROD model. FG domains coat the walls of transport channel saturated with NTRs, leaving an inner tube in central channel. The small molecules can diffuse through this channel, whereas the larger ones would be restrained. NTRs containing transport complexes interact with the FG NUPs at the entrance of the NPC and start a random two-dimensional walk along the wall of the central channel, possibly displacing cargo-free NTRs from their binding sites. The periphery of the channel is used at the preferred transport area in this model. Besides nucleoporins, which serve as an architectural scaffold and a permeability regulator of nuclear transport, other factors also affect transport across nuclear membrane. Caryopherins are a group of proteins involved in transporting cargo between the cytoplasm and the nucleus of a eukaryotic cell. Caryopherins can act as importins, that is, they help in importing proteins into the nucleus, or exportins, that is, help in exporting proteins out of the nucleus into the cytoplasm. More than 20 caryopherins have been reported in humans. Among them, 11 caryopherins, including importin beta, are involved in transport or bidirectional transport of cargo molecules, and 8 exportins involving exportin T are involved in the export of cargo molecules. Each caryopherin carries its own specific cargo molecule. Caryopherins can also help to export tRNA, miRNA, rRNA, viral RNA, and uridine-rich small nuclear RNAs with or without ribonucleoproteins as an adapter. In this picture on the left, you can see the structure of importin beta. The N stands for amino terminus and C for carboxy terminus. Importin B is composed of 19 helical repeat motifs, also known as heat repeats. Each consists of an A and a B helix connected by a short turn, which in heat 8, as seen in this picture, is replaced by an acidic loop, critical for the regulation of substrate binding and release. The heat repeats 1 to 8 are required for high affinity binding to RAN GTP. The importin beta binding or IBB domain of importin alpha interacts mainly with the residues located in repeats 7 to 19 of importin beta. The binding site for nucleoporins of the nucleopore complex is located between residues 152 and 352, corresponding to repeats 4 to 8. On the right, you can see importin beta in yellow color forming a superhelical structure that wraps like a snail around the IBB domain, which is represented in blue. GTPases are enzymes that hydrolyze guanosine triphosphate GTP into guanosine diphosphate GDP and release energy. A small RAS-related GTPase, RAN, regulates the ability of both importins and exportins to transport their cargo. RAN is in a different conformation depending on whether it is bound to GTP or GDP. This picture shows the RAN cycle. RAN cycles between its GTP and GDP bound form dependent on its subcellular localization. The different forms of RAN confer directionality to nuclear transport by deciding where caryopherins bind or release their cargoes. In its GDP-bound state, RAN binds to caryopherins, importins, and exportins. Importins release cargo upon binding to RAN GTP, while exportins must bind RAN GTP to form a ternary complex with their export cargo. The nucleotide binding state of RAN depends on its location. In the nucleus, RAN GTP is present, while RAN GDP is present in the cytoplasm. The association of RAN GTP with exportins moves the complex out of the nucleoplasm. When associated with importins, it leads to the release of cargo molecules. RAN GTP is about 100-fold more abundant in the nucleus than the cytoplasm, which is possible due to the presence of RAN's guanine exchange factor RCC1 in the nucleoplasm, but RAN GTPase activating protein RAN gap in the cytoplasm. A number of proteins and other raw materials of various sizes and dimensions are imported into the nucleus. As nucleus does not possess the function of translation machinery, that is, protein synthesis. Two major types of signals have been identified for the nuclear import of proteins: SV40 type and bipartite type. SV40 type was first found in the large T antigen of the SV40 virus. This type of signal is characterized by a few consecutive basic residues and in many cases also contains a proline residue. The bipartite type was first identified in Xenopus nucleoplasmin. Its characteristic pattern is two basic residues, 10 spacer residues, and another basic region consisting of at least 3 basic residues out of 5 residues. In this picture, you can see how nuclear localization signal on protein binds importin and helps to import the protein into nucleus through the nucleopore complex. The signal for nuclear export is a leucine-rich domain which can be recognized by a class of exportins called exportin 1. Some proteins which have to shuttle between the nucleus and the cytoplasm contain both nuclear localization signal NLS as well as NES, which is the nuclear export signal. Examples include the Rev protein of HIV-1 and the nuclear factor of activated T cell NFAT. The Rev protein plays a key role in the regulation of viral expression. NFAT is the target of an immunosuppressive drug widely used in organ transplantation. Cyclosporin A and FK506 act as powerful immunosuppressants by blocking the activation of calcineurin phosphatase activity, thereby suppressing activation of NFAT and NF-kappa B in T cells. This picture illustrates the nuclear export mechanism. tRNA export depends on the various modifications it undergoes, thus preventing export of improperly functioning tRNA. This quality control mechanism of nucleopore complex is important and ensures tRNA's central role in translation, where it is involved in adding amino acids to a growing peptide chain. The tRNA must mature inside the nucleus before export. Therefore, tRNAs are aminoacylated and only tRNAs charged with an amino acid are exported efficiently. The tRNA exporter in vertebrates is called exportin T. It binds directly to its tRNA cargo in the nucleus with the help of RAN GTP. The left part of this picture shows nuclear import cycle with cargo in orange and importin in red color. Important proteins bind their cargo in the cytoplasm and interact with the nucleopore complex to pass through its channel. Once inside the nucleus, interaction with RAN GTP causes a conformational change in this importin that dissociates it from its cargo. The resulting complex of importin and RAN GTP then moves back to the cytoplasm through the nucleopore complex, where a protein called RAN binding protein, RAN BP, separates RAN GTP from importin. This allows access to a GTPase activating protein, GAP, that binds RAN GTP and induces the hydrolysis of GTP to GDP. The RAN GDP produced binds to a nuclear transport factor which returns it to the nucleoplasm. Inside the nucleus, the RAN GDP interacts with the guanine nucleotide exchange factor GEF, which replaces the GDP with GTP, which again makes RAN GTP, and a new cycle begins. The right part of the picture shows nuclear export cycle. In nuclear export cycle, cargo is to be delivered to cytoplasm is marked in bright blue color and exportins are marked in dark blue color. Nuclear export is roughly the reverse of the import process. In the nucleus, the exportin binds the cargo and RAN GTP and diffuses through the nucleopore complex to dissociate in the cytoplasm. RAN GTP binds GAP and hydrolyzes GTP. The resulting RAN GDP complex is restored to the nucleus, where it exchanges its bound ligand for GTP. Therefore, importins depend on RAN GTP to dissociate from their cargo, whereas exportins require RAN GTP TP in order to bind to their cargo. This movie summarizes what we have discussed till now. It shows a eukaryotic yeast cell nucleus with its nuclear membrane studded with nuclear pore complexes shown as purple barrels with octagonal symmetry. You can see the donut-shaped nuclear pore complex is made up of 3 lattice-like ring structures: the cytoplasmic ring, the nuclear ring, and the inner ring complex. The 3 rings are sandwiched between cytoplasmic extensions extending 35 nanometer into cytoplasm and nuclear extensions extending about 60 nanometer into nucleus, and are linked by a distal ring to form a basket-like structure. About 30 nupes are present. Most important among these are the Fg nucleoporins with disordered symmetry. They interact with Caryopherins to transport the cargo across the nuclear pore complex. The NPC is a dynamic structure. You can very well appreciate the movement of cytoplasmic extension and nuclear basket between cytoplasmic and nuclear rings. There is a vertical gateway called central framework. This central framework harbors the central transport channel that accommodates all active transport across nuclear pore complex. The Caryopherin transports cargo from the cytoplasm. This Caryopherin cargo transport complex, shown in yellow, docks to the entrance of central transport channel with the help of cytoplasmic filaments or cytoplasmic extensions. The Caryopherin cargo transport complex moves inside the nuclear pore with the help of interactions with the Fg repeats. You can see how the Caryopherin cargo complex is interacting with the forest of Fg nups inside the channel of NPC to pass across it. The next step is the movement into the nuclear basket. This is facilitated by the release of RAN GTP. Following this, the cargo is successfully transported to the interior of the nucleus from the cytoplasm with the help of nuclear pore complex, Caryopherin, and RAN GTP. After studying the nuclear transport in detail, let us now understand how abnormal nuclear transport can affect cell function. Abnormalities in the nuclear transport can cause various disorders that involve protein aggregation, biosynthesis, or cell metabolism. In cancer cells, exportin 1 plays a critical role of exporting various tumor suppressor proteins including RB, retinoblastoma protein, APC, adenomatous polyposis coli protein, P53, P21, P27, and so on and so forth. Overexpression of exportin 1 has been associated with various cancers including acute and chronic leukemia, multiple myeloma, sarcoma, melanoma, glioma, ovarian, pancreatic, gastric, and cervical cancers. Mutations of nuclear localization signal can also alter cell function. When nuclear localization signal is mutated, a protein intended for nuclear transport from cytoplasm fails to reach its destination of activity or function, leading to profound problems in cell function. Let's talk about Swyer syndrome, a disease caused by mutation of nuclear localization signal of sex-determining region Y, SRY gene. Swyer syndrome is a sex reversal disease of males caused by XY gonadal dysgenesis. SRY is transported by a specific important to the nucleus to activate testis-related genes. SRY gene is responsible for formation of testis in Y part of XY chromosome. The nuclear localization signal mutation of SRY gene inhibits the absorption of testosterone and male organs fail to develop. The individual is born as a female with female voice, vagina, and fallopian tubes, but rudimentary uterus and no ovaries. To correct genetic or nuclear abnormalities, delivery of therapeutic DNAs or proteins into the nucleus has been successfully done by researchers. Researchers have inserted short nuclear localization signals like SV40 or HIV-TAT to increase the efficiency of the nuclear delivery. In this picture, you can see different methods of delivery of corrective DNA into the non-dividing cells using Caryopherins like importance and nuclear localization signal. These methods have increased the transfection efficiency of the target DNA. The insertion of nuclear localization signal has also shown great results for the delivery of CRISPR-Cas gene editing system. In the future, we may be able to develop an ideal drug delivery system for therapeutic drugs by using the nucleocytoplasmic interaction proteins and pathways. I will again summarize by saying that nucleus is the control center of the cell. The nuclear envelope is a physical barrier between nucleoplasm and cytoplasm. This nuclear envelope is a phospholipid bilayer membrane consisting of two layers, inner and outer membrane, separated by a perinuclear space. Nucleus exports materials to cytoplasm including mRNA, tRNA and ribosome subunits, and imports enzymes, nucleotides, regulatory proteins, etc. from the cytoplasm. The nuclear membrane is perforated by nucleopore complexes, which fuse the outer nuclear membrane and the inner nuclear membrane together and creates channels for nucleocytoplasmic transport. Nucleopore complexes, also known as NPC, are multi-protein suprastructures that provide channels for nucleocytoplasmic transport. NPC consists of about 30 types of nucleoporins or NUPs present in multiple copies consistent with the 8-fold symmetry of NPC. The donut-shaped NPC is made up of 3 ring-like structures called cytoplasmic ring, inner ring complex, and nuclear ring. Between cytoplasmic and nuclear rings is the central transporter or framework structure, which harbors a central channel through which all the active transport across the nucleopore complex takes place. The NUPs are organized in small set of subcomplexes which are defined by the stability of protein interactions. The largest subcomplex of the NPC is the Y complex. The factors affecting nuclear transport include nucleoporins, RAN GTPases, Caryopherins, nuclear localization signal and nuclear transport signal. Abnormalities in the nuclear transport can cause various disorders that involve protein aggregation, biosynthesis or cell metabolism, including several cancers. If you have any further questions, feel free to email me. I would like to wish you all the best for future learning. Thank you.